Hello students and welcome in today's session of DNA fingerprinting. Now DNA fingerprinting means, what do you think that? Is it related with our fingerprints? And what do you think, where the fingerprints of these our hand palm fingerprints are there, where it will be used? Yes, you must know the Aadhaar card. Yes, and whenever it is linked, this is linked with your fingerprints, right? And this will gives you unique identity. So what it proves that no two personalities will have the same fingerprints, right? It is not here any relation between these fingerprints and the DNA fingerprints. It is totally different. But yes, whenever we see the DNA fingerprinting, this technique is utilized to find the differences between the two persons, because no two persons are exactly same. Their DNA is never exactly same except the twins. So whenever we will see the DNA fingerprinting, in this technique DNA is matched of two different persons, and this matching of the DNA and finding their differences is known as DNA fingerprinting. Actually, this process is very complicated process, and you know that DNA is a very long DNA. We have seen that in Human Genome Project. It is not possible that to compare the DNAs of two different personalities at the same time and find the difference. So here we can find the differences in their DNA, and that differences we find. Now you can see here red color 99% bulk genomic DNA is given, and this white color 1% is there, that is of a repetitive DNA. So here first you understand that what is mean by bulk DNA. You already know that in our human beings total 99.9% DNA is the same DNA. It is no any change, and this fragment is there, bulk is there DNA, which is repeated thousands of times, right? And that big segments of the DNA which repeated for thousands of times forms the bulk DNA, and very small segment of the DNAs will be there which will have repetitions, but their size is very small and they have a differences in them, right? And they will form your repetitive DNA, which is a 1%. Even you have studied in Human Genome Project that less than 2% genes are there that code for protein. So here whatever the bulk genomic DNA is there, that will not code for protein. So this is what we are utilizing here, this 1% repetitive DNA for DNA fingerprinting. Now, just now I said that in 1% DNA that fragments are repeated many times, right? So you can take here an example of genome 1, genome 2 and genome 3. So these are the three different genomes, and here if you see the sequence, here sequence is GATA, and the same sequence is repeated here again and again continuously. So here if you will see in genome 1, it is repeated 1, 2, 3, 4, 5, 6, 7, 8 times, right? Even in the second genome also it is repeated 8 times. But if you will see genome 3, it is repeated 9 times. One extra is here. So if you compare this repeated sequence of the genome, it is varies in a different location and in different organisms, and this is what is known as tandem repeats, and this is what actually the base is considered for DNA fingerprinting. This repeated sequence are there this will be taken at a different loci from the different chromosomes, and which help us in DNA fingerprinting. Now you see here, this satellite DNA and main DNA is there. From these, whatever the different bands are there from the different chromosomes, if we consider this, then from that we are getting two different bands of the DNA after density gradient centrifugation. Now you know that density gradient centrifugation is the process where the DNA will be get separated based on their density. The heavy density will be settled down at the base and lighter will be at the up. And as you know, the bulk DNA is there, having a very big strands of the DNA forms the main bands which is settled down, and the repetitive DNA is there which makes the satellite bands which will be get separated because they are the smaller segments. Now if you see their graphical presentation, then in the graphical presentation you will see a small peak of a satellite DNA and a very big peak of a bulk genomic DNA. So in this way the satellite DNA is separated from the bulk genomic DNA by the density gradient centrifugation. Now in this satellite DNA there are difference in their length of the segments, maybe of a 70 nucleotide base pair is there, or maybe of 100 base pair nucleotides are there, or very maybe small or more than that long, right? So based on the length of the segment of that, then the base composition means AT rich segment is there or GC rich segment is there. Based on that composition and a number of tandem repeats, number of repetitive units, how many times it is repeated in the organism, right? Suppose that segment is repeated for 15 times in me, maybe in my daughter 10 times, or maybe in your it will be repeated for 50 times, or in someone's it may be of 100 times, right? So these numbers will be varies personality to personality. So based on these 3 points, length of the segment, base composition and a number of repetitive units, satellite DNA is divided into two parts, mini satellite and microsatellite. Now mini and micro, you know the difference between these two. Mini means small and micro means smaller than that. Now these differences are there which are based on their size. Now if you will see here, mini satellite, it will be larger than the microsatellite, right? So if you will see the size of the nucleotides in this, then it will be more than 9 nucleotides segment will be there, and size will be near about 1 to 5 kb size, right? So this is about the mini satellite, and it will be repeated for so many times, right? And microsatellites, if you will see, as it is of more than 9 nucleotides structure is there, then this will be smaller than this, means automatically it will be 1 to 8 nucleotides segment will be there, right? And size again it is of 1 to 5 kb, so it will be less than that. So in this way satellite DNA is divided into two parts based on their length segments and number of nucleotides. Now in a mini satellite DNA, you know that it is having more than 1 to 8 nucleotides base sequence is there which is repeated so many times, right? So this is what a structure you can see on the chromosome, and in this is what a very VNTR region. VNTR region means variable number of tandem repeats. Tandem repeats means so many times it is repeated, and these repetitions will have some of the variation. Now these variations will come due to mutation. Now how many these VNTR probes are there in a chromosome? So that will be varies in number, right? In a microsatellite, it will be less in number, but if you will see in a mini satellite, it will be more in a number, right? So in this way, these mini satellite and micro satellites are differ in the VNTR probes. Now, just now I said that these VNTR probes are there or VNTR segments are there, they will have the differences, small differences in them, right? Now, these differences are there, these will be create polymorphism in the population. Now, what is meant by polymorphism? So, this will have chromosomes, majority of the portion will be the same, but very small portion is there where a change is there. So this polymorphism you can see in the chromosomes, it will be occur. And whenever this polymorphism occurs in the germ cells, so we have two types of germ cell. One is sperm and another is the egg. So whenever this polymorphism change or mutation occurs in these two germs or any one of the germ cell, then definitely these two are taking in the process of fertilization. That's why this will be automatically transferred into the next generation. So this polymorphism goes on transferred into the next generation, and based on that polymorphism or based on that VNTR, DNA fingerprinting is carried out. Now this fingerprinting is carried out by Southern blot technique. Now this Southern blot technique is there. This is discovered by Alice Jeffrey. This is the scientist who has given this technique of Southern blot DNA fingerprinting. Now in that technique, what happens? See here, in Southern blot hybridization will be carried out. Hybridization will be carried out of what? This is what the target DNA, whose DNA fingerprinting we are doing. This will be digested with restriction endonuclease enzyme. Means it will be get separated, break down into the small segment, right? And these digested DNA is there. It will be run on the gel electrophoresis technique. Now this will be a gel shape on which the DNA material will be inserted, and it will be, we will see this ahead in detail. Just see here the steps. Gel electrophoresis it will run, then it will be transferred to a membrane or a nitrocellulose paper. Same here, some of the probes, known sequence DNA is there. Their fragments or probes will be made. These probes will be run on this nitrocellulose paper. So wherever their matching sequence is there they will get hybridized, and if it as it is a radioactive probe, it will seen in a radiograph photo. So this will be seen. See now one by one in detail. This is the first step where we will select the DNA, then isolation of a DNA. You know the isolation of a DNA, we have practical on that also. So in isolation of DNA, you take any material or from which we have to take out the DNA, right? So here we don't have to take a particular material only, because DNA is present in each and every cell of the human body, right? Or any organism. Then only one cell is again sufficient for a DNA fingerprinting. So you have to take out the DNA from that nucleus, right, by digesting the cell membrane and digesting the nuclear membrane, digest all the proteins, RNA, and by using the different enzymes, and you will get then pure DNA. So this is what the isolation of a DNA. Once you isolate the DNA, it will be very long DNA, that is 2.2 meter length of the DNA is there, right? So we cannot use that much long DNA as it is. So we need to cut it into the small fragment. So for that we need to use scissor, right? And do you know which is the biological scissor? Biological seizure is nothing but the restriction enzyme. So endonuclease enzymes will be used here to digest the DNA. So DNA will be cut into small fragments. Now these all fragments will be not of the same size. Some will be small, some will be medium size, some will be very large, long, right? So different fragments will be formed of the DNA. So this is what the DNA fragments. Now we have to separate these DNA fragments with the help of gel electrophoresis. Now this is the gel sheet. Here one cavity will be formed which is called as a well, right? So these two samples will be put into this cavity. As you know that DNA is negatively charged, right? So this end of the gel electrophoresis will be attached to the negative charge, and this end of the gel electrophoresis will be attached to the positive end, right? And means this is the negative end, this is the positive end. And even the DNA is also negative. Whenever we'll on the current, then the circuit will be complete and the current will flow. So automatically the DNA is a negatively charged. So it will start to flow towards the positive charge, right? Another thing is that this gel sheet is there. This gel sheet is made up of agar medium, right? So this agar is there. It makes this gel sheet as like a sieve, right? All of you know that what is mean by sieve? Yes, chani. Whatever we are using at home in kitchen to clear the floor, right? So from that unwanted material will be large size get separated. So these sieves are there. They will have the small size pores, and through that smaller than that pore size will be get filtered down and larger particles will be remain up. So the same technique is used here in the gel electrophoresis. So here the size of the pores will go on decreasing. So here it will be of a big size pore, then it will be smaller than this, then here again smaller than this, then again smaller than this. So the smallest will be at the base, largest will be at the top. So when we pour the DNA on this gel electrophoresis, when we pour this DNA in the well on a gel sheet, then DNA segments will start to percolate. Now whichever the long segment is there, very large fragment is there, it will be get separated here only, and smaller than that will be there, that will go to the next stage. Then according to their size and according to the size of the hole, the DNA fragments will get separate at a different level. The smallest size of the DNA will become at the last. So in this way the different fragments of the DNAs will get separated over here. As we have here two different samples, we are getting the separation of the DNA in the two different rows. So this is how we get separated fragments of the DNA, right? Now the next step is that these DNA fragments are there that will be transferred to the synthetic membrane. Actually it is called as a nitrocellulose paper, right? So this is what you can see a paper. When we will keep this paper on this and keep a weight on that, press it, then these all DNA fragments will be transferred on the nitrocellulose paper. So you can see here the paper is little bit torn, so you can see here the DNA's fragment get transferred on this paper, right? So this is what a transfer of DNA fragments on the nitrocellulose paper. Next thing is that hybridization of the DNA fragments using radio labeled VNTR probes. Now before this you have seen what is mean by VNTR probes, right? So variable number of tandem repeats. Now we have to do these VNTR probes hybridization process with these DNA fragments. Now, before doing this, we have to prepare the DNA which is of our own known sequence, means we know the sequence of the nucleotides in that DNA. The DNA will get separated, means single stranded. Then again the same restriction enzyme will be used to cut that pieces, so that will be cut it into the small fragments, right? And most important thing is that we are using here radioactive VNTR. Radioactive VNTR means the DNA which is formed is having a radioactive material in it, right? Now why we are using this radioactive material? Because whenever we will do this X-ray film, that time it will help us to locate their position. Now these radioactive VNTR probes are there, that we will spread on this nitrocellulose paper, right? So what we think? This is also single stranded and these probes are also single stranded. So because of that, these wherever complementary base pairing is there on this segment and this probe, they will get attached with each other and from a double stranded structure, right? So this is what having the same complementary structure on the DNA. So once this hybridization is done, then we can keep an X-ray film on it. So this is what a X-ray film. So this is X-ray film and take a photograph, autoradiography, and this will help us to detect the hybridized DNA by this autoradiography technique, right? So after autoradiography, we are able to see their film in this way. So these are the locations where these known fragments are having the same sequence in their DNA. As here we have taken the samples of two individuals. This is individual 1 and this is the individual 2 of this DNA sample, and this individual 1 is having this DNA sample. If you will check their probabilities or matching their probes with the DNA, it varies, right? So in this way we can see there is a difference in their genetic material of two individuals. This is what a DNA fingerprinting. You can see here if two different individuals are there, it will be varied. But if you do this for monozygotic twins, then it will be the same. Except monozygotic twins, it will be different for any two unrelated individuals or related individuals also. It will be not same for any two individuals other than monozygotic twins. Now, if you want to increase the sensitivity of this DNA fingerprinting, then you can do a Pcr technique. You can use the Pcr technique. Now, what is mean by Pcr technique? PCR, polymerase chain reaction. Now, what is this polymerase chain reaction? It will make a polymer of the DNA and it will be formed in the chain repetitively, and from the one DNA you can make billions of copies of the same DNA by this technique. So you can increase the segments of the DNAs in billions times by using this polymerase chain reaction. Just how it will be done, in short I will say, suppose this is a double stranded structure, get separated. Once it is get separated, add a primer, then this primer will go on extending. Second strand will be formed by adding complementary strand. On this strand also you add a primer, this will again go on extending and a new DNA strand will be formed. So in this way, from the one DNA, two DNAs will be formed. Again these two DNAs will be heated, break, hydrogen bonds open. So 4 strand we are getting, from that 4 strand 8 DNAs, and in this way go on increasing. So in this way, if you will do 30 cycles repetitively, you will get billions of the DNAs from the one DNA. Now this DNA available in some of the dispute also. Means wherever sometimes parenting problem is there, or if there is a dispute for the biological father, that time also this DNA fingerprinting technique it is utilized. Now how it is utilized? Same here, sample we are collecting, probes we are producing which is known, and based on that we will do. Another thing you see here, this is sample A, sample B for DNA fingerprinting, and if we will do this wherever we have a doubt, for that two persons you have to see, this is a mother's, this is child's DNA fingerprinting or the patches of the DNA, and this is the alleged father 1, and this is of 2. And if you will see, as a child will have the genetic material 50% from the mother and 50% from the father. So the child, if it is matching its DNA pattern or DNA fragments with the father, then you can say this is the child's biological father. This patch of the DNA is not matching with this father, right? So that's why we can clearly understand here the biological father of this child is this and not one this. So in this way, this is the first application of DNA fingerprinting. Another thing is that this is also used to identify the criminal. Now how is it? You must have seen the CID serial, right? And you also know that how our police works to find out the criminal. So whenever they visit any crime place, that time they are trying to collect each and every small proof, right? Whatever they got, the samples they will be collect. Maybe in that sample they will collect hair follicle or a small skin or mucus from the toothbrush or blood, traces of blood, drop of blood, maybe it is dried then also. Because for DNA fingerprinting a single cell is also sufficient, and as you know that every cell in the human body will have the same DNA content. So it doesn't matter that the cell has come from which organ, right? So that sample they will collect, and if they will have their more than 1% suspect and they don't know who is actually the criminal who is present on that place during the crime, right? So they will use this technique, DNA fingerprinting. So this is what the DNA material which we observe from the crime spot, and these are the 3 samples of the criminals. And if you will go on matching, which will have the exact matching with the sample which we obtained from the crime spot. So you can see this is the exact matching. Suspect 2 is exactly matching with this DNA fingerprint pattern. So you can say that the suspect 2 was present on that crime scene and he is the criminal. So in this way DNA fingerprinting is used for this also. So this is what DNA fingerprinting. Here we have finished this point, and even we have finished our chapter number 6, right? So this topic is very important. Now from the exam point of view, DNA fingerprinting is very important. I just now said the 2 applications of the DNA fingerprinting that will be asked. Another very important point of the process of DNA fingerprinting. This will be asked for 3 marks. Sub questions will be also asked on the of 2 marks based on the process of separation of DNA. Then what is mean by VNTR, what is mean by satellite DNA, bulk DNA, mini satellite, microsatellite. So these are some of the basic points, and based on that questions will be asked of 1 mark, 2 mark or sometimes 3 marks. 5 marks questions very rarely it will be asked, right? So from the exam point of view, you prepare this point very nicely. All the best.